Thank you very much, Anna, for your kind words. So, thanks for that very nice introduction. This is an area that you can apply in different ways, and a lot of this alludes to Mendelian randomization as well, as we heard in the previous talk. So the key point that I will try to address here, based on some studies, is what it actually is within diabetes, type 2 diabetes, that links it to hypertension and cardiovascular disease. Because we have to be honest and see that we have a lot of homework to do regarding prevention of cardiovascular disease in diabetes, because there really is no evidence that we can reduce the risk of type 2 of cardiovascular disease by lowering blood glucose, and there is no evidence that we can reduce cardiovascular disease by giving drugs that lower your body weight. And this is what is driving our studies, that we would like to find any other causal factors that links glucose metabolism together with cardiovascular disease beyond glucose. And I will hit down on four endocrine or three endocrine systems: natriuretic peptides, neurotensin, and vasopressin, and also some metabolites from metabolomic studies that we think are promising in this respect. So if we start with the natriuretic peptides, so this is how we classically view them. So when the cardiac pressure, or the pressure on the cardiac walls, increase, so natriuretic peptides will be released into the circulation. They will promote natriuresis and dilate blood vessels, and thereby reversing or attempting to reverse the subclinical or clinically overt overpressure in the heart. And if we look epidemiologically at the natriuretic peptides, this is data from the Malmo Diet and Cancer study. It is clear that in healthy subjects in middle age, that the higher circulating levels you have of natriuretic peptides, adrenomedalin, ANP, and BMP, they are all independently of all traditional risk factors, significant predictors of future cardiovascular events in healthy people. This is when it's important to think about these plasma biomarkers. What is actually the basal secretion of natriuretic peptide, the genetic secretion, and what is the compensatory secretion that we saw in the first slide? So we addressed this in a number of in Malmo cohort, Finnish cohorts, and Framingham Heart Study. And so this was really before the GWAS era, so we genotyped densely the AMP, BMP genetic locus. These genes are lying just between each other. And by doing that, we were able to really see a very strong association with natriuretic peptide levels, with some variants within the genes coding for the peptides. And as you can see here, with increasing with the alleles that went with higher levels of AMP and BMP, those alleles, those genotypes, were in sharp contrast to what traditional epidemiology says, associated with lower blood pressure. So genetically higher level of natriuretic peptides is associated with lower blood pressure, in contrast to what we see in traditional epidemiology. So really, and this has been replicated several times. So we think that it's fair to say that a genetic AMP deficiency actually is causally related to hypertension. And what is then the link with metabolic changes, the diabetes-related angle of this? So again, we worked with Framingham and Malmo Diet and Cancer Study, and first looking at natriuretic peptides and their relationship with obesity and insulin resistance. As you can see, for these, should we say, less pressure-dependent traits, you already see an inverse association between the plasma levels of natriuretic peptides and these traits, so that the higher levels of natriuretic peptides you have, the less likely you are to be obese, the less likely you are to be insulin resistant. And this is seen in both cohorts, a little bit stronger in the more obese American cohort than in the Malmo, but highly significant. So suggesting again that the deficiency of natriuretic peptides could be dangerous not only for hypertension but also for obesity and insulin resistance. We then moved ahead and related AMP in this case to prospectively in people with perfectly normal glucose values and no diagnosis of diabetes, to later development of diabetes in the Malmo Diet and Cancer Study, where which was followed up after 15 years with oral glucose tolerance test. And we also, of course, recorded any diagnosis that may have been made during this journey over 15 years. And here you can see that if you had a low levels of AMP here, you are at increased risk of developing diabetes later after adjustment for everything, including glucose within the normal range. So that those who have higher levels, they are protected from diabetes in comparison with the lowest quartile. And if you turn it around and look at this 25% with the lowest AMP values, so they have about 40% increased risk of developing diabetes later in life, adjusted for all confounding factors. So that's still at the plasma level. The first Mendelian randomization evidence came from Nick Wareham's group, where they associated they again showed that variation at the natriuretic peptide gene locus was strongly associated with levels of the peptides, but then took that on to using Mendelian randomization to show that the allele that goes with lower or with higher levels of natriuretic peptides will exert protection, again, a highly significant protection against diabetes. So this was the first evidence of causality between high levels and protection from type 2 diabetes or low levels of peptides and risk of diabetes. We are doing the same thing in the MDC and see the same thing, that the SNP that we have found strongest associated with AMP levels. So those people who have low AMP, we already show they have higher blood pressure, but they also have a higher risk of developing diabetes during long-term follow-up compared to the other genotype, which goes with lower blood pressure and higher AMP. So there are additional evidence also linking AMP deficiency to left ventricular hypertrophy. But this is the way we look upon it today, that you are born with, if you're unlucky, you're born with genetic AMP deficiency. And this will first lead you into insulin resistance and obesity, then diabetes and hypertension, and the onwards journey in this direction. But the compensatory secretion, of course, of the natriuretic peptides will increase during the way, so that for diagnostic purposes, the plasma levels may be high get higher at a certain time point in life where compensation exceeds the basal genetic secretion. So let me move on to example number 2 of where we think there may be a link between something that can link diabetes and hypertension and cardiovascular disease together. And this comes first from one of the genome-wide association studies of coronary artery disease. I'm not going to go through all this, but there's one player that had one of the strongest signals for risk of coronary artery disease and myocardial infarction. This is the sortilin-1. So sortilin-1 came up early as a myocardial infarction and CAD-associated gene. And in another collaboration study, we could dissect the mechanism that by which this allele causes MI. So a little bit what Jens was asking for. So the allele, which is located, the risk allele is located far outside of the coding regions of SORT1, but introduce a CEBP enhancing binding site. So if you have one variant SORT, that will increase the expression of SORT1, and to make long story short, sortilin-1 will go in and block the export of VLDL out into the circulation, where that's the protective variant. If you have the other variant, you will be have less blocking of lipid export out in the circulation, in the end leading to higher LDL cholesterol levels. And because, as has been shown very clearly, because LDL is causally related to MI, this will explain the association between SORT1 and MI. But certainly it was interesting to understand more what is the regulation of sortilin-1. And sortilin-1 doesn't only control the granule transport of VLDL, but also many of the glucose transporters, for example. So it's also implicated in diabetes. And so it turns out that this sorting protein, it's also a receptor, and it's a receptor for a hormone called neurotensin. So SORT1 is the same thing as neurotensin receptor number 3. And neurotensin is shown, it's a satiety hormone. So if you inject it in the brain or in the abdomen of animals, it affects the satiety, how much these animals eat. It also has effects on pain sensation, antinociception, and stimulation of growth. So, and there are 3 different receptors for neurotensin. This is the one that is identical to SORT1, the neurotensin 3 receptor. So when we measured neurotensin in the fasting state in about 5,000 people in the Malmo diet and cancer study, we found that there was a very clear interaction in this study between the levels of neurotensin and gender on the risk of getting cardiovascular disease. So that although in the population there was a highly significant increased risk with higher levels of proneurotensin as measured, this was entirely driven by females, and within the male subset there was really no association at all. What about diabetes? Because, as I told you, the SORT1 is implicated in glucose transporter trafficking as well. And because it is a satiety hormone, it's also likely that it could affect diabetes risk. And similar to the Cardiovascular Disease Association, so neurotensin predicted strongly new onset diabetes during follow-up, but only in women, not in males. And this effect is so strong that it affects how long people live. So people with the highest, or women, again only in women, with the highest levels of neurotensin, they have an increased risk of dying early. And this is, although neurotensin also, but I won't go into details of that, is also affecting risk of breast cancer. So the deaths seem to be driven by cardiovascular deaths that are associated with neurotensin. So from this gene and the ligand of SORT1 neurotensin, we have found another link that seems to be important in women. And why is it only in women? That's not known, but it is well known that from animal studies that you need to have a long window of estrogen exposure in order to produce to have neurotensin produced in cells. And and also women have significantly higher levels than males, also in our own cohort. So we believe there is an interplay with estrogen also because neurotensin was linked to breast cancer. What can you do something about this? We don't know that either, but we are doing studies on that at the moment. What we know is that the most effective way to get higher neurotensin is to eat a lot of fat. Acute studies, there is a clear increase of neurotensin. Whether reduction of fat intake will turn out to lower neurotensin on the long term and to lower the risk associated with neurotensin is yet to be proven or disproven. So the second endocrine system that we believe is important in linking diabetes with cardiovascular disease is actually the vasopressin system, which you know best from its renal effect on aquaporin production and regulation of water reabsorption in the kidney. But there are a lot of animal studies also implying a role of vasopressin, mainly through vasopressin 1A and B in the liver, pancreas, and most importantly, I think, in the brain, because vasopressin, it's been shown that stimulation by the vasopressin 1B receptor can lead to a Cushing-like phenotype, so that overstimulation by vasopressin on the V1B receptor in the brain, you would based on the animal studies expect to lead to increased secretion of ACTH and abdominal obesity and other features of Cushing. So with this, we got interested in vasopressin. We measured Copeptin, which is the C-terminal fragment of the vasopressin pre-pro hormone, and just for stability reasons, just like N-terminal BMP and AMP. And we found some years ago now that elevation of vasopressin is strongly predictive of new onset diabetes, and its predictive value is higher. The more even within the normal range, the more normal glucose you have, the stronger is the association between high vasopressin measured by Copeptin and diabetes. And not only is it linked to diabetes development, but coming back to the Cushing-like phenotype with hypertension, low-grade inflammation, and obesity, in particular abdominal obesity. We examine whether variation of the human V1b receptor gene would lead to some Cushing-like phenotypes, just like in the animal studies who were genetically manipulated. And we have found a very strong replicative association with obesity, overweight, or abdominal obesity primarily. We still owe you replications of the diabetes association, so we're working with other cohorts with no results yet, but it looks promising with that actually the V1b receptor is associated not only with obesity but also with diabetes. And to get back to the link with cardiovascular disease, this is the first time I have seen a biomarker that predicts strongly hard coronary endpoints and death, but only in diabetes patients. So if you have high Copeptin, it's a strong indicator of future risk of cardiovascular disease and total mortality if you're diabetic. But within normoglycemia, there is no predictive value of Copeptin or vasopressin levels. So if you want to do something about your high level of vasopressin, what would be the most and easiest way to get vasopressin down? That's definitely to drink more water. So we are now doing an intervention study which is not ready yet. Maybe I can get back to present this data some other time, where healthy people drink 3 liters of water extra per day, and we would like to see, we already know what will happen to vasopressin, but we want to see if this also can affect their glucose tolerance. And we are quite hopeful that there will be an effect, because on the animal experimental side we have shown that the insulin resistance that is seen in the obese sucker rat, you can ameliorate that insulin resistance of the rat by feeding that rat with high water, so the rats get a water-containing gel that they eat, leading to suppression of AVP. And the insulin resistance of the obese rat improves almost to the level of the lean rat. So this makes us hopeful for the human study to show some effect as well. So vasopressin, we also believe, may be a link between diabetes and cardiovascular disease. Final example is from the metabolomic studies, where obviously metabolomics, another omic, is very interesting in the way that it is kind of an interface between what you eat and how you take care of your food, your inherent and partially genetic inherent metabolism. And about 2 years ago, we did another collaboration with Framingham. So these are data from both Framingham and Malmo, and found that out of the global metabolome, so 3 branch-chain and aromatic amino acids, isoleucine, tyrosine, and phenylalanine, they were the clearly strongest diabetes-associated, or not prevalent diabetes, but for incidence of diabetes in the future. And it was very similar results in the 2 cohorts, both Malmo and Framingham. So the obvious question was, may these amino acids be associated with cardiovascular disease as well? And it turns out that clearly they are not only associated with atherosclerosis measured in the carotid artery, but actually predictive of future cardiovascular disease events in the Malmo diet and cancer cohort, the same 3 amino acids. So why do you get high levels of these 3 amino acids? That's still to be found out. But what we know based on the diet data in the MDC is that people who had reported, based on the dietary assessment, to have a lot of animal protein, they have clearly higher levels of these three diabetes and cardiovascular disease associated amino acids, whereas people who ate much milk and dietary protein, they had lower levels of these three amino acids. So we are now designing another intervention where we will, in a crossover design, have healthy people to have high milk protein and high meat protein sources, and see first if we can affect these diabetes and CVD associated amino acids, and secondarily, again, in the first as the first endpoint, whether that high milk protein could ameliorate glucose tolerance. And there is also genetic contribution. We have one for having high levels of these diabetes and cardiovascular disease associated amino acids. They are all strongly, so their levels in the fasted state are all strongly associated with a gene called MLIP. And this is a muscle A-type laminin interacting protein. So these laminin interacting proteins, they are highly expressed in heart, skeletal muscle, and smooth muscles in humans. And there is a monogenic disorder or loss of function of this gene, which causes dilated cardiomyopathy and lipid and muscular dystrophy and early aging. So we believe it has an interesting phenotype to be further dissected, and may be part of the function of how these three amino acids may be of relevance to human disease. So in conclusion, so when we integrate genome data with plasma biomarkers and risk, so we found that the AMP, BMP genes strongly affect AMP in the circulation. Can we do something about? Can we elevate AMP? Well, we can lower the salt intake. We will have more AMP left when it needs. And as you know, there are AMP elevating drugs. So maybe we can do something about that AMP deficiency as well. For neurotensin, and again, that neurotensin is a satiety hormone that stimulates one of its receptors, which is this coronary artery disease associated gene product. We don't know, but we think that maybe low fat intake will be one treatment. There are drugs blocking neurotensin 1 primarily, also neurotensin 2 receptor that are used for other indications. So there are potentials of doing something about the neurotensin excess risk as well. Vasopressin, I told you, high water intake. We'll see if that works. There are also developed V1b blockers that are available. And finally, the metabolites, can you do something about them? Well, that would probably mainly be through dietary interventions. Whether it works, we will see. So I'd like to acknowledge a lot of people in my group and collaborators close to my research group. Also to their American friends, in particular to Andreas Bergman, who is sitting here. Andreas was the person who developed 3 of the biomarkers I have been talking about, Copeptin, original AMP, and pro-neurotensin. So with those words, thank you very much for your attention.